going to go ahead and get started. I have a couple of announcements first off. The first is if you are not getting announcements about this seminar series, please, and you want to, please be sure to go to the SEM website and get on the mailing list because we have a lot of wonderful speakers this fall and of course, also in the spring. Secondly, if you are interested in participating in the Evolutionary Medicine Journal Club, it's held every other week on Tuesdays from 10:30 to 11:30. And so if you would like to participate in that, please shoot an email to Randy, who is organizing it, and he will add your name to the list so that you find out about the papers that will be discussed. Okay? Now, with that, it is my great pleasure to introduce Caitlin Pepperell. She is, let's see here. Okay, so she has her MD from Queen's University in Canada, and she then did, I guess, a residency in internal medicine and in an infectious disease specialty in Toronto. Next up, she had an infectious disease fellowship and research postdoc at Stanford University. And currently, she is an assistant professor in medicine and medical microbiology at the University of Wisconsin Medical School. So, welcome.
Thank you. Awesome. Are we good with the mic? Okay, sweet. Well, thank you so much for the invitation. It's, as a physician and an evolutionary biologist, this is a really special place for me to be. So thanks for the invite. I'm going to talk about tuberculosis, which is a deadly disease that is a strong interest of mine, both from a clinical and an evolutionary perspective. So the story of tuberculosis is still being written. I'm going to give you a completely biased version based on how I think about this bug. So full disclosure there. I've got a picture of a library here because the stories of microbes are, in my opinion, more interesting and more diverse than boring old species like humans. So humans have one story, whereas pathogenic microbes, we're really talking about a library of stories in terms of their emergence and their persistence in human populations. So I'm going to focus on TB. And to introduce you to this character, I'm going to put it in context with some of the other pathogenic species of bacteria because I think it's helpful, in order to understand what TB is, it's helpful to have some context and understand what it isn't and how it compares with other pathogenic species of bacteria, because in many respects it's quite singular. So the first up in terms of comparison is Staphylococcus aureus. This is a bacterium that lives in the nose of a large proportion of us. Something like 20% of people carry it in their noses, and it doesn't do much of anything there. However, Staph aureus, when I'm on clinical service at the hospital, is at the top of the list of bad guys that I deal with. So it lives in the nose and it doesn't do much of anything from our perspective, but then can emerge in a virulent form and cause bloodstream infections, brain infections, bone infections, etc. And the preprint that's referenced here is a really cool study looking at adaptation of virulent Staph aureus from this commensal reservoir. So in the case of Staph aureus, we can think of disease and pathogenicity as an adaptation, an emergent property from this resident community of bacteria. This contrasts with tuberculosis in that tuberculosis is much more dedicated to being a bad guy. So TB, unlike Staph aureus, cannot be transmitted in its commensal or latent state. TB must cause disease, it must cause tissue damage, it must cause mischief in order to be transmitted. So it is an obligate pathogen in a sense that is not true for most of the organisms we think of as bacterial pathogens. So the next comparator is a bug called Clostridium difficile, and here we have an empty classroom. And this image refers to the fact that the disease that's associated with Clostridium difficile is as much about the absence of things as it is the presence of this really somewhat, in some sense, pathetic bug. So we can think of Clostridium difficile associated diarrhea, which is a terrible and worsening problem, as with a very crude but yet useful model in which the normal, happy, healthy bacteria that colonize our gut are impaired or depopulated, and this nasty critter emerges. So in this case, pathogenicity and disease has to do with an emptiness, in a sense. This contrasts with MTB, which does not colonize the gut. And in fact, nowadays it's out of fashion to say that there are any sterile tissues in the human body, but MTB occupies as close to sterile as we get. So it's really not a part of the primordial slime that we have in our throats and our guts. It's deep in the lung tissues and really living in isolation from other bacteria. Klebsiella pneumoniae is another emerging health threat and a super fun bug. Melissa and I were talking about species concepts with bacteria, and this is a really good one to think about in that respect, because it's essentially a modular collection. Its genome is a modular collection of stuff from all over the place. It occupies a vast niche space. There's klebs in all kinds of environments, and they readily exchange bits of modules of DNA, and when the right modules come together, it can form a pathogen. So broad niche space, lots of exchange of DNA between strains. Again, a very strong contrast with MTB, which has a very narrow niche space, and as far as we can tell, completely clonal evolution. So I've looked very, very hard for evidence of recombination, and I wish I had found it, but I haven't. So as far as we know, perfectly clonal. I like this image, which is MTB bacteria in orange meeting a human, binding to receptors on a human macrophage, because I think it very nicely illustrates the intimate association of Mycobacterium tuberculosis with its human host. So it is operationally a human-restricted pathogen. It is an obligate pathogen. It's clonally evolving. It's in many ways what we classically think of as a bacterial pathogen, something that we can wrap our arms around. But I just wanted to make you aware that the world of bacterial pathogens is incredibly complex, and our understanding of it is itself evolving. So as an obligate human pathogen, the story of tuberculosis is intertwined with the story of humans. It would be, I think, a waste of time to consider MTB evolution completely in isolation from humans and what we are and what we do and how we hang out and what our history is. So chapter 1, Emergence of the Monster. This is just one of my favorite topics, and I think of interest to many people. Most bacteria are innocent and irrelevant as far as we're concerned. There is this tiny proportion that like to murder us. And it's nice to know, what are the evolutionary and ecological mechanisms that lead to the emergence of the pathogens we know and the ones that we're going to encounter in the future. So going back in the history of bacterial pathogen evolution, pretty early on, it was observed that the diversity of Helicobacter pylori appeared to be shaped by ancient human migrations. So these observations were made in inferences, hypotheses were developed in the pre-genomic era and are now quite well developed. And there's a really beautiful literature on evolutionary genetics of this organism that I would encourage anyone who's into this kind of stuff to check out. Anyway, in my opinion, the story with Helicobacter pylori was quite misleading as far as MTB is concerned, because it led to a parallel hypothesis in MTB. So about 10 years ago, this was the original description, and then followed up here, presented a hypothesis that perhaps MTB emerged in Africa 70,000-ish years ago, that the structure of bacterial populations reflected ancient migrations out of Africa and reflected the structure of the genetic structure of human populations and subpopulations, and even that human populations and MTB populations have co-evolved. And so the idea here is that the strains of MTB that are found in East Asia, for example, would be specifically adapted for East Asian human subpopulations. So this is the hypothesis that's been around for some time now. So people use the term co-evolution. It gets bandied about a fair bit. And I thought it would be useful to sidestep and talk about a definition of it that I like that was published some time ago. That's not elegantly worded, perhaps, but I think captures the important concepts. So an evolutionary genetic change in a trait of the individuals in one population in response to trait of individuals of a second population, followed by an evolutionary response by the second population to change in the first. So back and forth, reciprocity. It follows from this that the two populations should be evolving over similar timescales. If their rates of adaptation are wildly different, it's hard to see how they could reciprocally evolve. And it should be evident as codivergence in the phylogenies. So the timescales over which MTB evolves have been estimated numerous times. They were summarized in a paper that I published last year with Vegard Eldholm from the Public Health Institute in Norway. And if you want to dive into this literature, the references are listed here and they're in this paper. So the rate of TB evolution has been estimated from epidemiological and historical data with very concordant results. These are the different lineages. So some evidence that lineage 2 might have evolve at a higher rate than lineage 4. And then we have 2 ancient DNA studies, which have slightly lower rates, which is expected based on our understanding of how rates evolve over time and looking at different depths of divergences. And then the out of Africa, the rate that you would have to invoke in order to support the out of Africa hypothesis is here, which is clearly an outlier, and in my opinion, for which there's little support. So MTB timescales do not appear to be similar to human evolutionary timescales. The data from the ancient DNA studies points to an emergence, a time to the most recent common ancestor of less than 6,000 years before present. Maybe Monica and I were talking about maybe it's 8,000 years, but not 70,000 or 80,000 or 90,000, as has been suggested in the literature. And I want to just make a sort of a point that often gets lost here, in that the time to most recent common ancestor is not equivalent to the time that tuberculosis emerged as a disease. So because TB is clonally evolving, it's possible and even likely that the population has been subject to clonal replacements, where a single clone displaced ancient diversity. So I personally think that that's very likely, and that there was some ancient diversity prior to 6,000 or 8,000 years ago that we just simply have lost. Another possibility is that the disease has emerged on multiple occasions. So there are some, you would know better than me, older archaeological historical data suggesting that a disease like TB has been around for longer than that. And there are all kinds of what I think are even more fun possibilities to think of in terms of how that could operate, in that, perhaps pathogenic mycobacteria have emerged from environmental mycobacteria on more than one occasion. The disease patterns are not totally specific, so maybe it was a completely different organism that we know nothing about that clinically mimicked tuberculosis. And finally, I say here there's no evidence of codivergence between human and TB populations, and this is a little bit saucy because I didn't find evidence of codivergence, but my competitor did, but my method was better, so you should believe me. Okay. And if you want to know the references, so my analysis is in here, and then the competitors is there if you want to look at the details. Okay, so this leads me to the Pepperell hypothesis, which is possibly wrong, but I think useful. And also I really like it because it's not probably testable, which is nice. So my hypothesis is, or idea is that it makes a lot more sense to think about MTB co-evolving with human culture than with humans. I think the timescales are much better matched for how human culture evolves than sort of how we evolve in the sense of things like our out of Africa migrations. So in support of the Pepperell hypothesis, I'll tell you about some work that I did looking at tuberculosis in Western Canadian First Nations and imprints of human culture on MTB evolution in these populations. And Jeff and I were talking about genetic drift, and this is a story about different flavors of genetic drift. So pictured up here is a TB transmission network. So each of the dots is a person with tuberculosis disease. And then the arrows are inferred transmission events based on contact tracing data. So I had tuberculosis, you had tuberculosis, we live in the same house, we were named in this database, so then you end up as dots and lines on this network. So this is from a high incidence TB community, or a community that experiences high TB incidence in Western Canada based on contact tracing data and clinical information. And I hope you can see that the dots are different colors. So these are largely red, green, and these are blue. And the colors of the dots correspond to the individual's community of residence. So these are 3 or actually 4 different communities, cases in these different communities. So zooming out, you can kind of see that there's structure in terms of, who transmits to whom. The reds tend to transmit to other reds, and the greens to other greens. So this simply makes sense. Humans don't assort and mix with each other randomly. And lo and behold, this is reflected in terms of who we infer you transmit disease to. So this is based on clinical and contact tracing data. This study, published in MBE, was looking at the bacterial population. And I was interested, yeah.
Just really quickly, what's the time frame?
For this?
So how long does that take?
This was over a few years. I honestly can't remember how many, but it would sort of be in the 5-year range. Okay, so moving over to the bacterial side, I was interested in structure in the bacterial population, the scale over which the bacterial populations are subdivided from each other, and what different explanatory phenomena we could find to explain variation. And so we looked at simple things like isolation by distance, the First Nations affiliation of the person, their ethnic affiliation, a number of different schema, and looking at basically a sort of a nested model, building up from community to agency to region. We found a very high degree of structure, and particularly at the level of, this is a historical government term, by the way, Indian agency, particularly at the level of Indian agencies, where 28% of the bacterial genetic diversity differentiation could be explained by comparisons between agencies. And this is over a pretty small scale, so the agencies would encompass a few thousand people. The agencies are really interesting structures. So these are government structures, and they have a historical meaning. They were set up at the time that the Canadian reserve systems and band affiliations were set up. And they reflect both intrinsic organizations, because the government structures reflected people's family and household and cultural and nation affiliations. It also reflects the structures that were imposed on these communities. So it reflects, where people were settled as far as reserves. It reflects how government resources were distributed. And it reflects, probably most importantly, things like residential schools, the organization of residential schools. So both factors that were intrinsic to the populations, human populations, and sort of structures that were imposed from outside. So that's population structure. Another flavor of drift that's very common in MTB populations are founder effects. And for those of you who are not population geneticists or don't think about this all the time, you can think of it as an organism, in this case TB, landing on really fertile soil. So there's nothing particularly special about the bug, but it just happens to be in the right place at the right time. And as a result of this, leaves a lot of descendants in the population. So the study that I published here that I referenced before, one of the things we looked at was sorting communities into those that had early onset epidemics and those that had later onset TB epidemics. And these are all related to disease ecology issues. So essentially to things like food insecurity and institutionalization of the populations. So both of those factors are very promoting of epidemic forms of TB. And those phenomena were not, the different communities didn't experience them all at the same time. So some experienced severe food insecurity and broad institutionalization starting in the late 19th century. And for some communities, it was delayed until even the 1950s and 1960s. And we could see the impact of this in terms of the diversity of the bacterial populations. So the communities with younger epidemics had less diverse bacterial populations versus those that had historically more mature epidemics. In subsequent work, I looked at migration of MTB across Canada via the Canadian fur trade. And found evidence suggesting that the dispersal of MTB, so movement from A to B, and the expansion of MTB populations can be asynchronous. So it appeared as though the migration events occurred first, and the bacteria were propagated and supported in these populations at low levels, and then bloomed in later periods in the face of conducive ecological pressures such as crowding and food insecurity and famine. We estimated the demographic history of the global MTB population and inferred a 25-fold expansion, which is shown, the timing of it is shown here, which is coincident with a huge expansion of human populations, as well as probably important things like urbanization and colonial migrations. So to summarize, MTB is an obligate human pathogen. Transmission varies by environment. So as I said, situations of crowding and starvation and famine are highly conducive to the transmission of TB. And the genetic diversity of MTB populations there's some evidence that is shaped by the social architecture, what I call social architecture of the human host populations in terms of the structure of contact networks, historical trade, and colonialism and its myriad attendant impacts. And what's shown here is a residential school from Fort Resolution, Northwest Territories, where we know from historical data that the transmission of TB was, I would use the word extraordinary. Huge problem among many. So switching gears from Canadian First Nations and to the global scale, one of the interesting questions that comes up when we think about a shallower time scale for the bacteria that we're dealing with today is if it didn't get around the world through human migrations out of Africa, how did TB spread? Because TB is found everywhere in the world today. And so it's sort of a fun question to try to understand how that happened, and probably relevant to some things too. So the project I'm going to present now was done by Mary O'Neill, who was a graduate student in my lab and is now a postdoc at the Pasteur Institute. What she did was reconstruct the migratory history of MTB in the Old World using whole genome sequence data from about 500 isolates. And the migratory history was very complex. There were a lot of really interesting things that came out of the analysis. One sort of really fascinating to me side note is patterns with China and Southeast Asia. So China is host to a very large proportion of the world's TB population. It's obviously a very big country with a lot of people in it. And the rates have gone down quite precipitously, but are still high enough and historically have been high enough that, I don't know, if you counted all the MTB bacilli in the world, China would host a large slice of the pie. But China has been quite irrelevant in the history of dispersal of MTB, which was sort of surprising to me. Southeast Asia emerged as much more prominent a player in the spread of TB around the world. And China really was important in its exchanges with Southeast Asia, but has not been a big player in the spread of TB. And I think a historian perhaps would make an argument that historically isolationist policies in China may have contributed to this pattern. So, as I said, MTB is clonally evolving. It's also divided into 7 distinct sublineages. And because of its clonal evolution and because of this, I think, because of the strong population structure, the lineage structure is very well maintained. And when we looked in detail at the data, each lineage essentially had its own story as far as its migratory history, its demographic history, and the relationship between spread and growth of the bacterial populations. So we're also interested, of course, in what historical context might apply to these reconstructed migration events. And we applied the rates from the ancient DNA papers. We didn't try to estimate the rate of MTB evolution, but we just applied those rates and then enlisted the help of colleagues from the Department of Classics in Madison, William Aylward, and then Alex Zarley is a student of both classics and geography to help us make sense of the possible historical context for these migration events. And clearly, when you reconstruct a tree and migration and estimate timings, you can come up with some historical justification for it and sort of make a just so story. So I don't want to oversell, how confident we are, but just make the point that when we map MTB migrations in space and time using this time frame, we have very sensible historical phenomena that would have led to shifts in connectivity between populations and that fit the data qualitatively very nicely. Monica and I were talking about one example, which are trading ships that crossed the Indian Ocean in the context of the maritime spice trade, and then later Roman trading ships, which, looking at the pattern of the bacteria, seem to have been a really important dispersal tool for tuberculosis. And our historian colleagues tell us the way they describe the ships, they certainly sound as though they would have been very conducive to transmission of TB. Alex told me for some odd reason that on the Roman ships, the people working on them were nude while they were on these ships, which seems like cruel and unusual punishment. But anyway, just a side note. Don't say I didn't teach you anything. All right. Okay, so I've been talking about the bacterial population. But in our co-adaptation hypothesis, we need to also think about human culture and how MTB and other disease agents have, what kinds of impacts they've had on human cultural evolution. And for the most part, I would leave this to historians and sociologists who can bring a rigorous framework to it. But I want to just tell one story that came out kind of surprisingly in some of the epidemiological research going on in the lab. So this project was done by Erin Zwick, who is a mathematician, and she's working on TB transmission models and quite sophisticated models and complicated models. And my vision for her first project was like a very unsexy pedestrian paper where we would estimate try to estimate disease duration. And so these kinds of papers, even though they're maybe a little bit boring, are really nice because they're useful basically for anyone doing TB transmission modeling to have some estimates of the parameters that go into the model. So I had some data from Western Canadian sanatoria. And again, looking at First Nations patients. And looking at the admission date and discharge date for individuals admitted for TB treatment. And the data set, interestingly, covered the period over which, straddled the period over which TB drugs eventually became available. So the first drugs became available in the late 1940s, and then multi-drug therapy started to be used in the early 1950s. And the data set went from the 1930s to the end of the 1950s. So we really just wanted to get an estimate for how long people are sick with TB. And then also I was interested in how this might have changed as drugs became available. Naively, before Erin did the study, I would have thought that the availability of drugs would have shortened people's duration of treatment, their duration of disease. Certainly with the drugs that we have available now and the regimens that we have available now, that would be true for the majority of patients. There are, of course, always people who are very acutely and severely ill who would die quickly. But for the majority of patients who have untreated TB, it's a disease that plays out over months and years. And what we found was, or what Erin found was, in fact, the opposite. So the length of stay in the sanatoria actually increased over time, and particularly there seemed to be a threshold effect where it increased following the availability of effective chemotherapy. So I think in understanding this, we can go back to the concept that, well, perhaps the TB treatments didn't work the same way that they do now, and maybe this is just the natural history of treated TB in this time period, which could be. I also think it's useful at this point to bring in the perspective of medical historians who have, there's some really wonderful medical historians who've studied these populations and particularly looked at how TB control measures, TB treatment measures were applied to First Nations communities in comparison with white populations. And they make the argument that once institutional structures were built, so once the sanatoria were built, once the nurses and doctors were hired, and this whole infrastructure was developed to control and treat TB, that there was an impetus to maintain those structures. So once you had built the hospitals, to fill the beds. And this is a phenomenon that's familiar to healthcare economists. And they argued that the way that the First Nations, the way that this played out with First Nations and tuberculosis was essentially that because these treatments were so effective, they ran out of white people to treat. And at a certain point shifted, policies clearly shifted to applying TB control and treatment efforts to the First Nations communities. So I do wonder about a role of this sort of, we have the hospital, we have these beds available, we need to fill them, and we're going to keep people here for treatment in order to justify that as playing a role in sort of how this evolved over time. The final chapter in this story is eventually the policies shifted away from inpatient treatment, both for First Nations populations and white populations. It happened later for First Nations populations, but by the time we get to the early 1960s, there was a movement away from this type of intervention. Aaron looked at at why people left the hospital over time. And in orange are people who ran away from the hospital. So this is one of the codes for how people left the hospital. And then another code were those who were evicted from the hospital. So a substantial proportion of these hospital discharges were runaways and evictions, which I think speaks to the complexity of public health interventions and the fact that they may not, in fact, be unfettered goods. So that's a little bit about how human healthcare culture evolves over time. And how our ideas change about who needs treatment and why and how we're going to treat. And for the last vignette, I'll tell you a little bit about the reciprocal impacts of those kinds of phenomenon going back to the bacteria. So one of the really interesting things that came out of this study with Vegard that we published last year was an impact of institutional structures, specifically the collapse of the Soviet Union on adaptation of MTB. So Vegard looked at this Central Asian clade of MTB that's found in Central Asia, as the name implies, in Russia, and now also in Northern Europe, and mapped drug resistance mutations on the phylogeny in time and in association with various geographical locations. And when you do that, these map very nicely to the collapse of the Soviet Union and presumably the breakdown of public health infrastructures. So a direct impact of kind of how our institutions evolve and how that in turn can affect a whole population of bacteria and leave a problem with multi-drug resistance TB that we're still dealing with. So I'll just summarize there and say the stories of humans and MTB are intertwined, and I kind of trolled humans, but I really do like them. Most of the data point to a TMRCA in the past few thousand years. I think that reciprocal adaptation is more plausible for human culture than it is for humans. There are many examples of impacts of human social structures, social architecture on MTB adaptation, and bidirectional effects of TB on MTB evolution and human institutions. And I'll end with my acknowledgements, and thank you for your attention. Yeah?
Why is food insecurity so conducive for TB?
Do you want me to repeat the questions for the... Yeah. So the question was how food insecurity is conducive to TB, basically. So food insecurity this is really a fascinating topic. We just had an outside speaker in India, from India, come to Madison, and he has a vision for controlling TB in India through nutrition. So he sees nutrition as a TB control tool. And the reason for that, the reason that it's so potentially powerful, obviously it would be great to feed people who are hungry for many reasons, but TB happens to be a really good one. It changes the probability that you'll develop disease after you're exposed to TB. So right from the get-go, most people who are infected with TB will not get sick. If you're very malnourished, that's a huge, that's going to bump up your risk very significantly. It makes the disease, it's associated with a higher disease burden. So you're more likely to have a worse form of TB, a more transmissible form of TB. People who are malnourished do not absorb their drugs, their TB drugs, as well as people who are well nourished, and they experience more side effects from TB drugs. So it really is a multi-pronged effect. Yeah?
What is the signature of adaptation of the human genome against TB?
So the question is, what is the signature of human adaptation to TB?
Yeah, so you were talking about co-adaptation.
So
like, how do we know that there's co-evolution?
Right. So as far as I know, there's no signature of, or there's no sort of simple signature of adaptation in the human genome to TB. There are lots of people who are looking at trying to understand the genetic basis of susceptibility to TB. I was more arguing that human culture evolves in response to TB. Mm-hmm.
Very nice presentation. I like it very much.
Thank you.
What type of model did Erin use to measure the duration
and
in hospital?
The interactions, a linear regression?
And whether or not you use the same model with existing data now to see whether you have the same conclusions.
What's that?
Whether you use the same model.
Oh, for a different data set?
Current data set, yeah. To see if you have the same conclusion. Like what type of model? Is it compartmental type model or agent-based type model? Or?
So she's working on an agent-based model of TB transmission. What was presented there was a linear regression. So those were two separate projects. Yeah, and we couldn't ask the same question with modern data because we use drugs now. So we couldn't ask, whether the disease duration changed as an effect of, whether you were treated or not, because we treat everybody. So that would be hard to do. Any other questions? Yeah.
I just had a question about your estimate of growth rate, your 25-fold estimate.
Mm-hmm.
That was from allele frequencies, that estimation. Mm-hmm. Are you worried at all about accounting for purifying selection and those sorts of estimates? Because they're both just going to strongly left skew your frequency spectrum. And if you're not accounting for that, especially in a linked genome, then it's just going to look like outlandish growth because that's your only parameter you're turning to fit a left skew.
Yeah, so we estimated the strength of purifying selection at the same time.
So you used a DFE somehow in that inference?
Well, it's a single locus.
Uh-huh. So you had a single value for deleterious mutations. I see.
Yep.
Thanks.
Yeah. And the purifying selection was strong. It's hard to say what those parameters mean, but yeah. Mm-hmm.
Actually, I'm curious about something you said early in the talk about C. difficile.
Oh.
So I was wondering if people are using probiotics or yogurt or something to fill the niche before C. difficile can reestablish after antibiotic use.
They're using poop. So fecal transplants, yeah. I mean, sure, we always recommend probiotics for everything because they're fairly innocent as interventions, but the fecal transplants. Stan Falco, who's an eminent bacteriologist, wrote a piece in one of the ASM journals that I just read. It was a blog post about when he was training in the 50s, and there was a surgeon, a GI surgeon, who had this idea that people who had surgery and went on antibiotics, that their guts got messed up, to put it in technical terms. And so they harvested poop. Like Stan Falco harvested poop from pre-op patients, stored it, and then the surgeon gave it back to the patients after their surgery, and they'd had their perioperative antibiotics without telling them what it was. But anyway, it was a really interesting article. It's a long time ago. Yeah. So the idea of fecal therapy has been around for a while.
All right. Thank you very much.
Thank you.